Hey there, listeners. Welcome to another paper preview podcast. Credit for this work goes to the original authors. Please find a link to their paper on PubMed Central in the description below or using the QR code on screen. This paper preview is meant to be a general overview and introduction to the paper. Please read the actual text and figures for more details. The following discussion, script, and voices were created using AI and are not intended to mimic the likeness of the original authors.
Welcome back to the deep dive. We're here again to really dig into some cutting-edge science, pull it apart, and give you the core insights. Today, we're looking at a paper that's, well, honestly, it feels pretty significant for how we think about neurons keeping themselves healthy. It's this really surprising discovery in C. elegans, the worm, about how neurons handle stress. They eject these huge bubbles, basically called exifers. But maybe even more surprising is how these exifers then, interact with the cells next door. It's not just trash disposal. It looks more like a complex two-way chat that kind of challenges how we've thought about neurons and their support cells, like glia. The paper's called Large Vesicle Extrusions from C. elegans. Neurons are consumed and stimulated by glial-like phagocytosis activity of the neighboring cell. Quite a mouthful, but it delivers. So our mission today, especially for those of you deep in biomedical research, maybe doing your PhDs or postdocs, is to give you a really solid grasp of this cell communication. We want to pull out the key findings and what they might mean for your own research, possibly even sparking your ideas about neurodegenerative disease. And we're incredibly lucky today to have the lead author of this paper right here with us to give us the inside story. Okay, let's unpack this.
Thanks. Yeah, really happy to be here and talk through this work. Our main goal really was to go beyond just noticing these exifers. We wanted to understand, their whole life story. Where do they come from? What happens to them? And most importantly, what does it all mean for the neuron, especially using C. elegans as our model system? It turned out to be, way more complex than we first guessed.
Great. So, mind to kick things off, for listeners who aren't super familiar with this whole exifer thing, could you just paint a picture?
Yeah.
What are they exactly? And what makes them such a, unique and kind of extreme way for neurons to handle stress?
Yeah.
I remember seeing images. They're massive, right?
Absolutely. Yeah, massive is the right word. On a cellular level, they're giants. We're talking several micrometers across. I mean, sometimes an exifer can be nearly as big as the neuron's own cell body, the soma, where it buds off from. And they pop up when C. elegans neurons are under serious stress, what we call proteostress.
Right, proteostress. And just to remind everyone, especially if you're working in cell biology, that's when the cell's quality control systems for proteins get overwhelmed, right?
Mm-hmm.
Like you can't fold or degrade proteins properly anymore.
Exactly. That's spot on. And the stress can come from lots of places. You could block the proteasome or autophagy, the main recycling centers, or maybe hit the cell with oxidative stress, starve it, or force it to make proteins that tend to clump up, like the Huntington protein with that long polyglutamine tail, HTTQ1-A, which is, directly linked to Huntington's disease. So basically, when the neuron's drowning in toxic protein junk or busted organelles, it doesn't just, give up. It makes this huge membrane-wrapped garbage bag, if you like, and physically throws it out.
Wow. It really does sound like a sort of last resort for detoxification. A pretty dramatic biological move.
It really is. And our first idea, based on what others had seen too, is that this had to be protective for the neuron. Like, it's the cell's big attempt to just jettison a whole load of toxic stuff that its internal cleanup crews, the proteasome and autophagy, just can't cope with. And this kind of extreme cleaning is super important for neurons because, unlike a lot of other cells, they generally live a long time and don't divide. They can't just dilute their problems by splitting into daughter cells. They have to manage decades of wear and tear, protein turnover, all that stuff for the whole existence.
And that brings us right back to why this is so relevant for you listening, particularly if you're studying neurodegenerative diseases, because problems with this protein housekeeping, this proteostasis, are right at the heart of diseases like Alzheimer's, Parkinson's, Huntington's, where you see these protein clumps forming. So if neurons have this unique large-scale way to chuck out aggregates, figuring out how it works could be, huge. It might point to totally new ways to intervene therapeutically.
Absolutely. And for quite a while, the big question mark hanging over exifers was, what happens next? We could see them bud off, pinch off, and then what? Do they just drift off? Do other cells eat them? What happens to all the toxic stuff inside? That question, the fate of the exifer after it leaves the neuron, that was really the key thing driving the research we're talking about.
Okay, and this is where the story starts getting really interesting because as your work shows, it's not just about the neuron doing its own thing in isolation.
Precisely. So We first really got a handle on these things in C. elegans neurons. They sort of emerge from the cell body, the soma. For a bit, they stay connected by this really thin nanotube-like tether, and then, snap, they're released. And these things are packed, full of misfolded proteins. We often tag them with fluorescence so we can see them, and also damaged mitochondria. Basically, all the stuff a stressed neuron wants to get rid of. And our earlier studies had already shown pretty clearly that neurons that do manage to make exafers seem to function better and survive longer, especially if they're loaded up with something like that Huntington Q128 protein. So that backed up the protective idea.
That's really strong evidence for protection. But what's striking is, this isn't just some weird worm thing, is it? We see similar stuff elsewhere.
Exactly. That's what's so compelling. This seems to be a conserved strategy. Similar vesicle shedding has been seen getting rid of, say, oxidized mitochondria from mouse heart muscle cells, cardiomyocytes. There are also long-lived cells needing good quality control. And even more directly, large vesicles that look an awful lot like our C. elegans exafers have been spotted in human and mouse brains. And crucially, their numbers seem to go up in disease states, like in samples from Alzheimer's patients. That really suggests this isn't just a fluke. It's a fundamental biological process relevant across species, maybe even clinically.
That conservation is fascinating. It points to something pretty fundamental. Now, for your detailed work, you focused on specific neurons in the mechanosensory touch receptors, especially one called ALMR. Why that specific system? What makes it so good for watching this happen?
Yes. The ALMR neuron in C. elegans is, well, it's a fantastic system for this. First off, exafer production there is surprisingly consistent. We see it happen in about 10 to 20% of these neurons, usually around day 2 of the worm's adulthood. That specific timing helps a lot. And critically, it seems to be a one-shot deal for each neuron. It happens once, dramatically, and that's it. This predictability makes experiments and analysis much, much easier. But the real advantage is the worm itself. C. elegans is transparent. You can literally watch processes unfold in a living animal under the microscope. We could do time-lapse imaging, track fluorescently tagged cargo inside the exafer, and see the whole life cycle. The budding, the connection, the release, and then what happens next. This kind of live longitudinal tracking is incredibly powerful. It lets you see dynamics you'd miss otherwise. And one of the very first things we noticed doing this careful tracking was that almost as soon as these big exafers were released, they started to break apart. They fragmented into lots of much smaller vesicles. We called this the starry night pattern, or SN vesicles, just based on how it looked. And then after a while, these little starry night dots would also vanish. That fragmentation and disappearance really screamed active clearance to us. It wasn't just passive diffusion.
Okay, so the neuron does this massive ejection, the exafer pops off, full of toxic stuff, and then it breaks into these smaller starry night bits. But the big question is still there. What happens to them then? And what role did you find for the cells right next door, the hypodermal cells, which for our listeners are basically the worm's skin cells, right? How did you suspect they were involved?
Exactly. That was the next logical step. The ALMR neurons sit right up against this hypodermal layer, so it seemed like a good bet they might be involved in the cleanup. We started with really careful tracking, literally watching individual exafers over time in live worms using high-res time-lapse microscopy. We needed to know the timing, and what we saw was pretty consistent. Most of the exafers, marked with M-cherry fluorescence, did break down into those SM vesicles. And crucially, that fragmentation always happened before the M-cherry signal finally faded away completely. On average, the fragmentation happened fast, maybe 2 to 2.5 hours after the exafer first appeared. But the whole process from the exafer forming to the signal being totally gone took about 3 to 4 days, something like 80 to 98 hours.
Okay, that quick fragmentation followed by a relatively rapid disappearance, yeah, that really points towards an active biological cleanup, not just things drifting apart. Something is definitely doing something to them.
Precisely. And one really key early clue came from a small fraction, maybe about 7%, of exafers that just didn't fragment. They stayed big and intact, and they hung around for ages, well over a week, without being cleared. That failure suggested something was broken in the cleanup machinery, pointing towards an active, probably cellular process. So to pin down the hypodermis, we looked for telltale signs of phagocytosis, cell eating. When a cell engulfs something, it typically remodels its actin cytoskeleton and recruits signaling lipids like PI4-5P2 to the site of engulfment. So we made worms expressing fluorescent biosensors for these, specifically in the hypodermis, one for F-actin using uTRCH, and one for PI4-5P2 using the pH domain of PLC delta. And the results were striking. About 3/4, 74% of the newly formed exafers got coated in this dense layer of hypodermal F-actin, and around 65% showed distinct spots of PI4-5P2 P2s surrounding them. But importantly, the hypodermis right next to the neuron's main body, the soma, didn't show this actin or PI45P2 buildup. This told us the hypodermis was specifically recognizing the excifer as something to engulf, not just reacting generally to the neuron being there.
Wow, so it's a targeted response.
Yeah.
The hypodermis can tell the difference between the neuron itself and this package it just ejected. That's really specific cell communication.
It is. And our time-lapse movies confirmed this active engagement. We saw the hypodermal actin typically show up after the excifer started budding out from the neuron. It then quickly gathered around the bud, suggesting the hypodermis was reacting dynamically to the excifer's appearance. Then, to nail down if actin was actually required, we used RNAi to specifically knock down actin and related proteins just in the hypodermis. We targeted different actin versions, ACT1, NAX2, NAXA3, and a key part of the ARP23 complex, ARCS2, which helps build branched actin networks needed for engulfment. And in every case, knocking these down led to a big pileup of large intact excifers and far fewer of the starry night vesicles. So that sealed it. Actin-driven phagocytosis by the hypodermis is absolutely essential for getting rid of these excifers and breaking them down.
Okay, so the hypodermis is definitely eating these excifers using a standard actin-based mechanism. Now, once it's engulfed, what's the journey inside the hypodermal cell? Does it actually break down that toxic cargo, and how does that whole process, phagosome maturation, unfold?
Right, so once engulfed, the exifer is inside a phagosome, which needs to mature to fuse with lysosomes for degradation. We first looked at the large, intact excifers just after engulfment. They showed only pretty weak and transient signals for early markers like RAB5, or later markers like RAB7, or even the autophagy marker LGG1, which is like mammalian LC3. It suggested maybe some early interactions, but not full-blown processing at that stage. But then, when we looked at the starry night vesicles, the fragments, it was a totally different picture. Much clearer signals. These smaller vesicles strongly lit up with a whole bunch of maturation markers. We saw PI3P, an early phagosomal lipid using the 2XFYVE sensor. We saw RAB10 involved in recycling. Consistently, we saw the late phagosome Mendesa marker RV7, and really significantly, they were strongly positive for LGG1LC3, suggesting fusion with autophagosomes is a major part of their processing route. This pattern strongly indicated that the SN vesicles are the maturing phagosomes heading for destruction. And our electron microscopy backed this up. We saw these smaller, multi-layered vesicles around 500 nanometers in the hypodermis near where excifers were being made, fitting the size of these fragmented phagosomes.
Interesting. So it's like the big excifer gets engulfed, maybe sits there a bit, then fragments, and those fragments are what really go through the full maturation pipeline, including autophagy involvement. What about the lysosomes, the final disposal unit?
Yes, lysosomes are key for the final breakdown. Interestingly, the large intact excifers didn't seem to acquire much LMP1, which is the worm version of the lysosoma marker LMP. This actually made sense if degradation is fast, you might not catch the fusion easily in normal conditions. So to test this, we used a mutant called CUP5. QP5 is a lysosomal channel, and mutants have defective lysosomes. In these CUP5 mutants, suddenly we saw more LMP1 signal on the SN vesicles, and the vesicles themselves were bigger and held onto their mCherry signal more strongly, so that confirmed it. Lysosomes are crucial for degrading the cargo efficiently. But importantly, the CUP5 mutation didn't stop the initial fragmentation into SN vesicles. This tells us lysosome fusion is a late, fast step that happens after the fragmentation. We also dug into what drives that fragmentation. We looked at SAN111, which activates RAB7, essential for early to late phagosome maturation. Losing SAN1 caused a massive pileup of large intact excifers and almost no SN vesicles. This was huge. It directly linked that phagosome maturation step, activating RAB7, to the physical active fragmentation. We also found that mutants in ARL8, a GTPase that recruits motor proteins like kinesins, showed delayed fragmentation. So motor proteins are likely involved in pulling these things apart too.
Wow, okay, so it's a really orchestrated process maturation signals like RAB7 activation triggering fragmentation, possibly involving motors. You also implicated CNT1 and RAB35. How do they fit in?
Right. We knew PI455P2 was important early on, so we looked at regulators of phosphoinositide signaling. Two key players that popped out were CNT1, an ARF GFP, which basically switches off ARF GTpases, and RAB35, another small GTpase. These are often involved in membrane trafficking and phagosome maturation, sometimes by controlling ARF6 activity and PI45P2 levels. What we found was that mutants in CNT1 or RAB35 caused a really dramatic accumulation of the large intact excifers. They just weren't getting cleared, sticking around for days longer than normal. And crucially, putting CNT1 or RAB35 back only in the hypodermis fixed the problem. So their job is definitely in the hypodermis, facilitating clearance. We could also see tagged RB35 decorating the outside of these large exifers, and CNT1 forming little puncta nearby, supporting their direct role. They seem essential for efficient processing after engulfment.
This paints an amazing picture of the hypodermis as this super efficient cleanup crew for the neuron, engulfing, chopping up, degrading. It's impressive. But like you said, most of that describes the hypodermis reacting to what the neuron throws out. Your paper then dropped this, well, honestly, this bombshell finder. The hypodermis isn't just passively cleaning up, it's actively telling the neuron to make exifers in the first place. That just flips the whole script. So what does this mean? And how did you even figure out this was happening? It seems so counterintuitive.
It was definitely the biggest surprise of the project. So we'd just shown CNT1 and RAB35 were needed for clearance, lose them, and exifers pile up. And we knew from other studies they often act by inhibiting ARF6. So the logical prediction was, okay, lose ARF6 and exifers should also pile up, right? Same pathway. But when we looked at ARF6 null mutants, it was the complete opposite. Instead of accumulation, we saw a massive drop in the number of exifers being made, and hardly any starry night vesicles as a result. This was totally backwards from a simple clearance defect. It meant losing this phagocytic protein didn't stop cleanup. It stopped the production of the things that needed cleaning up. It suggested ARF6 was somehow influencing the neuron's ability to even make exifers.
Whoa, okay, hold on. So the hypodermis cell, this glial-like cell, isn't just a reactive janitor. It's actually involved in, instructing the neuron stress response, telling it to detoxify. That's a fundamental shift.
Exactly. It changes the dynamic completely. So we had to confirm ARF6 was acting in the hypodermis. We did rescue experiments. Putting ARF6 back only in the hypodermis was enough to bring exofer numbers back to normal. And the flip side. Using RNAi to knock down ARF6 just in the hypodermis copied the null mutant phenotype fewer exifers. So its crucial role is definitely in the hypodermis. We also saw tagged ARF6 accumulating in spots right at the edge of the forming exofer, suggesting it's right there at the interface, involved in the action. We even checked this in a DAF2 mutant background that worms normally make more exifers anyway. But even then, losing ARF6 still drastically reduced exofer numbers. It's a strong effect. Maybe the most revealing thing was watching time-lapse movies of DAF2 ARF6 double mutants. We saw neurons start to make little exofer buds, but then they often just retracted back into the cell. They couldn't seem to grow into full-size released exifers. This really suggested that hypodermal ARF6 is needed somehow to help that bud grow, or stabilize it, or prevent it from being pulled back in. It's actively facilitating the process.
So the hypodermis via ARF6 is almost like coaching the neuron, saying, go on, get that exifer out. I'm here to help. And you figured out how ARF6 fits in with RAB35 and CNT1 too.
Yes, we worked out the hierarchy there. Using genetics, doing double mutants, we found that the high exifer numbers you see in CNT1 or RAB35 mutants were completely erased if you also knocked out ARF6. So CNT1, ARF6, and RAB35, ARF6 double mutants looked like the ARF6 single mutant, very few exifers. This genetic suppression told us RAB35 and CNT1 must be acting as negative regulators of ARF6 in this pathway. And fitting perfectly with that, an ARF6 version that's always on a gain-of-function mutant actually caused more exifers to be produced, basically mimicking the effect of losing the brakes, RB35 and CNT1. So the model we came up with is RAB35 turns up CNT1 activity, CNT1 being an ARF-GKP, then turns down ARF6 activity in the hypodermis. So if ARF6 activity is high, either because ARF6 itself is overactive or because its inhibitors RE35-CNT1 are gone, then the hypodermis actively signals to promote exifer production by the neuron.
That's a beautifully worked out regulatory circuit. Amazing how these molecular switches control such a big cellular decision. Did you find any downstream targets, like what does ARF6 actually do in the hypodermis to send this signal?
We did look for effectors. We screened a bunch of potential ARF6 targets using that hypodermis-specific RNAi approach. One really interesting hit was SEC10. SEC10 is part of the exocyst complex. And you might know the exocyst is involved in tethering vesicles for secretion or membrane delivery. Knocking down SEC10 in the hypodermis looked a lot like knocking down ARF6 fewer exifers, fewer SN vesicles. This hints that maybe ARF6 and SEC10 work together in the hypodermis to manage membrane trafficking, perhaps delivering a recycled membrane to the site where the exifer is forming, and that this hypodermal membrane delivery somehow facilitates the neuron's ability to actually extrude the exifer. We also looked at PPK1, a kinase that makes PI435P2. Knocking that down was interesting. It didn't reduce the total number of exifers made, but it did cause more exifers to get still attached to the neuron, failing to release fully, and it reduced SN frequency. So this suggests PPK1 might act downstream of ARF6, maybe in a later step related to finalizing the engulfment or release. Perhaps ARF6 has an early role in promoting budding via SEC10 and a later role in contention involving PPK1. It's complicated.
This is just layer upon layer of interaction. It's incredible. So thinking about this complex two-way street, what about the classic phagocytic receptors, the ones we normally think of as just recognizing eat me signals like CED1? How do they fit in? Are they just doing the engulfment part, or are they also playing this surprising stimulating role like ARF6 seems to be?
Yeah, that was a really critical question, especially for CED1, which is the worm version of Draper and is so famous for clearing apoptotic cells. And what we found for CED1 was, again, really unexpected and super important. Losing CED1 in SEED1 mutants caused a significant reduction in both exofer and SN numbers. That phenotype looked remarkably like the ARF6 loss, or the SEC10 knockdown. And this was a key distinction, because for apoptotic cell clearance, losing CED1 causes dead cells to pile up uncleared. Here, losing CED1 meant fewer exofers were being made in the first place. This strongly argued that CED1, like ARF6, wasn't just involved in cleanup, but was actively involved in promoting exofer production.
Okay, wait, so CED1, the canonical EB receptor, is actually stimulating the neuron to make the thing it's supposed to eat. That really turns the standard model on its head, doesn't it? Usually the receptor just waits for the signal.
Exactly. It suggests a much more active participatory role for the phagocytic cell and its receptors than we usually assume, at least in this context. We confirmed CED1's role was in the hypodermis by putting it back just in those cells which rescued exofer production. And to see where it was acting, we used a version of CED1 tagged with GFP that hangs around a bit longer. We saw this CED1.GFP specifically lighting up around the periphery of the emerging exofer bud, but not around the main neuronal cell body. This implies CED1 is recognizing something specific on the exofer surface, some difference between the bud and the rest of the neuron, and this recognition is part of the trigger mechanism. So then we asked, what is it recognizing? What's the eat me signal here? A classic signal is exposed phosphatidylserine PS on the outer membrane surface, and CED1 can bind to PS-related signals. We looked at mutants involved in PS exposure like TR52 and NO1, and indeed losing either of these also caused a strong reduction in exofer production. So putting it all together, it looks like the neuron exposes PS or a related signal on the exofer bud. The hypodermis recognizes this signal via CED1. And this recognition, perhaps alongside ARF6 activity, actively promotes the neuron's ability to complete and release the exofer.
That is just elegant. A biochemical go signal from the neuron, read by the hypodermis, which then signals back, okay, keep going. A real feedback loop. What about CED10, the RAC homolog? Does that also stimulate production?
No, CED10 gave us the opposite result, clearly separating the roles. Unlike SEED1 mutants, SEED10 mutants are knocking down CED10 in the hypodermis led to an accumulation of intact exofers. Fewer SN vesicles formed. So CED10 loss looks like a classic phagocytosis defect failure to engulf or process properly. We also saw tag CED10 enriched around the exofer, consistent with its known role in driving the actin rearrangements needed for engulfment. So the conclusion here is pretty clear. CED10 is essential for the execution of phagocytosis after the exofer is formed, but it doesn't seem to influence whether the neuron makes the exofer in the first place.
Okay, that's a crucial difference. It really helps separate these functions. So based on all this, it sounds like you can group these hypodermal regulators into two distinct job categories, right?
Yes, exactly. We see two clear functional classes emerging from this work. First, there's class 1, the promoters. These are factors in the hypodermis that actively promote or stimulate exofer production by the neuron. This group includes ARF6, SEC10, the receptor CED1, and the PS exposure factors, TTR52 and ANOH1. If you lose any of these in the hypodermis, the main outcome is that the neuron makes fewer exofers. They are part of the signaling system that encourages formation. Then there's class 2, the executors. These are required in the hypodermis to actually carry out the phagocytosis process after the exofer has been formed. This includes the machinery for engulfment, fragmentation, and degradation, things like actin, ARP23, CE10, RAB35, CNT1, SAND1, AR8, and the lysosomal factor CP5. If you lose these, the main result is an accumulation of exofers or their fragments, because the cleanup process itself is broken. They handle the mechanics of removal, but don't seem to influence the initial production signal. Making this distinction is really key.
This is just incredibly detailed work, mapping out this whole intricate conversation between a neuron and its neighbor. Really changes the perspective from just waste disposal to active collaboration. Stepping back now, what do you see as the really big takeaways? What are the broader implications of finding this two-way street? especially for thinking about neuroprotection or tackling neurodegenerative diseases?
Well, I think the most direct implication is that it really solidifies the idea that exofer production is neuroprotective, but adds this crucial layer of external regulation. We show that if you block exofer production completely, like in the R6 or SED1 mutants, the neurons don't function as well when the animals get older. They had reduced touch sensitivity specifically at day 10, but not really at day 5. This suggests that this whole process, including the hypodermal stimulation, is important for maintaining neuronal health and resilience into old age when stress naturally accumulates. Now, okay, ARF6 and CED1 do other things too, we have to acknowledge that. But combined with our older data on the Huntington worms, it strongly supports a protective role for this active, regulated extrusion pathway.
So it's not just getting rid of junk, but the regulated process itself contributes to resilience over the long haul, particularly with aging. That's a really important point.
Exactly. Another interesting aspect is the fragmentation part, the starry night formation. This process of breaking down a large phagosome into smaller vesicles actually looks quite similar to what happens in other situations where cells engulf large things, like clearing dead cells in the worm embryo, or how mammalian macrophages handle big targets. So it suggests a conserved strategy for dealing with bulk cargo. But we also saw some unique features here, like the strong involvement of apophagy markers, LGG1-LC3, specifically on the fragments, and the fact that lysosomes seem to come in quite late after fragmentation. These details might differ from some other phagocytosis models and are interesting areas for future comparison. Perhaps the most conceptually exciting part is this instructional relationship, the phagocyte influencing the neuron. It really echoes other examples of glia-neuron communication, like glia pruning synapses during development, or even studies in flies showing transfer of protein aggregates from neurons to glia. It suggests this kind of active partnership, where the eater cell influences the ejected cell, might be a pretty common theme in biology. You even see it, as mentioned, with macrophages sticking close to heart cells to take up their exofers. It points to a fundamental need for close interplay.
So definitely not just a passive cleanup, but a real dynamic dialogue, critical for tissue health. That's a major reframing.
Absolutely. And it adds layers of complexity.
Right.
We also found that even within C. elegans, the roles of proteins like ARF6 and CED1 seem different in exofer clearance compared to, say, clearing the linker cell during male development. This tells us these pathways are highly tuned to the specific context, the type of cargo the cell's involved. You can't just assume a protein does the same thing everywhere.
Right. Context is everything. So this work has answered so much, but like all the best research, it throws open new doors. What are the big questions still bugging you? What do you really want to figure out next?
Oh, there are so many. A huge one, especially thinking about diseases. How completely contained is this process? Can any of that toxic stuff inside the exofer actually leak out of the hypodermal lysosomes? Could it reach the hypodermal cytoplasm or even get passed on again to other cells? That's obviously critical for understanding potential spreading of pathology versus effective containment. Another major question is the signal from the hypodermis to the neuron. We know ARF6 and CED1 in the hypodermis promote budding, but how does the neuron sense that? What's the molecular signal crossing that gap to tell the neuron, okay, go ahead and bud? That intercellular communication pathway is still a black box. And then there are finer mechanistic details, like precisely how are things like ER membranes or motor proteins like kinesins recruited during that fragmentation step. Nailing down the exact molecular choreography there would be fascinating. Answering these could really open up new ways to think about intervening. #tag outro.
Wow. What an absolutely fantastic deep dive. We've journeyed from seeing neurons under stress, throwing out these giant garbage bags exofers, to discovering this incredibly sophisticated partnership. The neighboring hypodermal cells aren't just passive cleaners. They actively stimulate the neuron to make these exofers, using complex signaling involving ARF6 and CED1. It's a highly regulated, collaborative effort, crucial for keeping neurons healthy long-term. This research really does shift our view of how cells talk in the nervous system. It's not just about what happens inside the neuron, but this active dialogue with its support system. So for you listening, as you tackle the challenges of neurodegeneration, protein clumps, and cellular stress in your own research, here's a final thought to take away. What if therapeutic strategies shouldn't only focus on the neuron itself? What if we could target these supportive phagocytic partner cells, not just to boost their cleanup ability, but maybe to enhance their ability to signal to the neuron, to encourage its own protective mechanisms like exophagogenesis? Could modulating the environment be a powerful, perhaps overlooked way to help neurons help themselves? It definitely opens up some completely new avenues for thinking about intervention. A massive thank you to our guest today for walking us through this truly groundbreaking work and sharing such fantastic insights. This has been The Deep Dive.
We hope it's given you plenty to think about. Until next time, keep exploring, keep questioning,